we'll be talking about Ulam distance, measuring permutation differences. There are different ways to measure differences in permutations, such as Kendall-Tau, Chevy-Chev, Hamming distances, but today we're talking about the Ulam distance. So for example, we could have two sequences. How different are they? You could have two DNA sequences, as we see here, that might be scrambled. You could have two different playlists that might be scrambled. So how can we measure this? Let's get to the technical definition of an Ulam distance. So the Ulam distance between two permutation arrays is the length of the permutation arrays minus the longest common subsequence of the permutation arrays. Remember that permutation arrays are just permutations of each other, so they're going to have the same length. Let's take a step-by-step example. So if you have these two permutation arrays, you could see that we have 1, 3, 5, 6 in the same order, and that's why we have the longest common subsequence as 1, 3, 5, 6, and that's length 4. The total length is 6. So the Ulam distance between these two is 6, which is the total distance, and then 4, which is the longest common subsequence, and that's how we get 2. What does that actually mean? That means that we just have to move 2 where it belongs and 4 where it belongs, and that's how we could have pi transform into sigma. And now let's get into the layman definition. So the Ulam distance counts the minimum moves to go from one permutation to the other. So in our previous example, we can fix pi by moving 2 elements. Why does this matter? It's good for block errors, not just single swaps or mismatches, and so it can be used for bigger errors. And it's also used in error correction for data stored as rankings like flash memory, and it helps us compare DNA and protein sequences in biology, and this actually helps us design bigger and safer permutation codes for the kinds of errors we expect in cryptography.